Our final ASN Young Investigator today is Nathaniel Sharp, and he'll be talking to us about the rates and effects of mutations are altered by multiple types of genomic variation. Nathan? They showed you how to...
Hi, everybody. So I obviously have to start by saying thank you very much to the ASN for the award. It really is a big honor to join the ranks of the young investigators. So I'll talk to you today about mutation. And you know, in a way, it's not that hard to explain why mutation is an important subject. People often say mutation is the ultimate source of all genetic variation. And that sounds kind of cool, and it's true. When we think about the major forces in population genetics, like selection, drift, and gene flow, those things actually aren't that relevant unless you have the genetic variation that's generated by mutation. So the other thing that people say most often about mutation is most mutations are deleterious when they have any effect at all. Many are also neutral. And again, that's true. The fact that deleterious alleles arise constantly because of mutation and have to be removed by selection means that there's this mutation load on populations. On average, individuals are less fit than they would be in the absence of mutation. Mutations are also causing other problems like inbreeding, depression, and negative health effects. Occasionally, we do see beneficial mutations, which are contributing to adaptation, of course, as well. So while no one would deny that mutation is important, we might often just assume that mutation is this background process that generates variation and makes this other stuff possible. And you know, I think we can be forgiven for doing that in a lot of cases, but what I want to try to convince you of today is that there can be some important variation in the causes and consequences of mutation. That variation can be systematic, and we don't yet entirely understand where it comes from or what its effects will be. But it's likely that it's going to have an impact on how we think about how populations evolve. So somewhat more specifically, I'm going to talk to you about how the effects of mutations can depend on a particular axis of genomic variation, which is sex, being males or females, as well as the effect of genetic quality and other kinds of genomic variation on the mutation rate. Before I go too much further, I want to acknowledge my PhD supervisor, Anil Agrawal, who played a very significant role in my career in general, as well as the work I'll tell you about today. This is the most fabulous picture of Anil that I could find. OK, so I'm just going to start with a little bit of theory here that'll help us think about mutation load in sexual populations. So let's just first think about a simple case of the mutation load at a given locus. So a deleterious allele will be present at some frequency in the population Q, and it has some effect on fitness S. We have a 2 here because we're thinking about diploids. And that equilibrium frequency of the deleterious allele depends on its rate of introduction by mutation and the rate that it's removed by selection. And so the rate that it's removed by selection and its effect on fitness are the same, and they cancel out. Oops. And so we're left with this classic result that the load at a locus just depends on the mutation rate. And this relationship tells us that across the many loci in the genome, mutation load can actually be substantial for a large number of organisms and drastically reduce fitness. But things are a little bit different if we're thinking about a population that has males and females. So now the equilibrium frequency of the deleterious allele depends on how strongly selection is acting against that allele averaged across the sexes. But its effect on fitness will depend on the sex you're thinking about. So let's think about the mutation load experienced by females. Now selection on females and the average selection across the sexes aren't necessarily the same. And so mutation load will depend on how strongly the allele affects females relative to how strongly it affects males. Female mutation load might often be the thing we're interested in if we're worried about population persistence, because it's really female fitness that's going to contribute to the productivity of a population. So what it means is that if selection on females is weaker than average selection across the sexes, the mutation load experienced by females will actually be reduced. So why might selection on females be systematically weaker than selection in males with respect to deleterious alleles? Well, one possibility is sexual selection. We often think of sexual selection as this process that might generate exaggerated ornaments, for example, and might act in opposition to natural selection, but there's no reason to think that it can't act on any locus in the genome. For instance, a mutant male might have a very difficult time finding a mate. On the other hand, a mutant female might have not much trouble at all finding a mate. So we might expect sexual selection to hit mutant males a lot harder than mutant females. But there was surprisingly little data on the sex-specific effects of new mutations when I started thinking about this, and so it's something I wanted to explore. I did this work in fruit flies, and so I wanted to get new spontaneous mutations to look at, and to do that I did a mutation accumulation experiment that I'll tell you about now. So this is the genome of Drosophila melanogaster, but for the purposes of this project, you only need to think about the second chromosome, which is an autosome. Okay, so we have one copy that's marked with BW, it's a recessive allele. The other copy marked with BG, a different recessive allele. I've highlighted one copy in blue there because that's the one we're going to focus on and follow through the experiment. If I take one male with this genotype and cross him to females with this genotype, a handy thing about fruit flies is that males have opted out of meiotic recombination and crossing over, so there'll be no genetic exchange in the male. There will be crossing over in the female. So the result of this cross is that, well, they have lots and lots of babies, but I'm just showing 4. Half the offspring will carry this chromosome that was the same one that was in dad, so the blue chromosome marked with BW there. So I'm just going to zoom that out a little bit. Now here's where the trick of mutation accumulation comes in. What I'll do is just select one male with that genotype at random and use him to sire the next generation. Okay, so we take that one male chosen at random and made him to, again, some females with these recessive markers. And each generation, choosing one male at random, that focal chromosome marked in blue will be transmitted intact from father to son. Except when a mutation occurs, nothing changes. So this is essentially like evolving a chromosome in the presence of mutation, but it's in the absence of selection. And the reason for that is that when we choose one male at random, we're essentially reducing the affected population size here so that selection can't effectively remove deleterious mutations. It's a kind of experimental evolution, you could say, where we're looking at just the effects of mutation alone without selection. So what I did was maintain many lines that look like this for about 50 generations or 2 years. Okay, so then once mutations have accumulated, I want to test their effects on males and females. First, I did some fly tricks to make those chromosomes homozygous. I can replicate them out. And then I put those genotypes, males and females, in a competition assay with some competitor flies of the same sex and flies of the opposite sex. And I essentially just score how many offspring they were able to produce. So we end up with quite a number of offspring, as I said, so we get 250,000 flies from an experiment like this to count, and that's a lot of fun. Okay, so we see really strong selection on adult sexual fitness in this experiment, and the effect of mutations was significantly stronger on males than on females. So from these data, we'd infer that the mutation load experienced by females is actually 20% lower than it would be in the absence of males. We also see a positive correlation in the fitness values for these mutation accumulation lines. And that's good to know about because it implies that mutations reduce fitness in both sexes, but more strongly reduce fitness in males. And so that extra selection that's happening in males will help to remove alleles that are also deleterious to females. So I've done some other work to try to understand the generality of that effect and other aspects of sexual selection and mutation load. And I don't have time to talk about these things today, but I do want to highlight that we still need more data on the sex-specific effects of individual mutations. I've done that a little bit. The mutation accumulation data, of course, multiple mutations are accumulating per line. It shouldn't be too much trouble to get more data like that, so I encourage you to go out and gather some, particularly if you're interested in studying something that's not a fruit fly for once. I also want to highlight that we need to understand how much of the variation in sexual fitness, the standing variation in a population, comes from things like deleterious mutations and their sex-specific effects versus alleles that might be under sexually antagonistic balancing selection. So I would conclude that sex is actually an important dimension of genomic variation that we should consider when we're trying to assess the selective impact of new mutations. We can't go out and measure the effects of a given allele on all possible genetic backgrounds a lot of the time, but we should at least try to do it in both sexes because this seems to be a pretty important dimension of variation. Sex-specific selection of this kind has now been observed several times in fruit flies as well as other lab organisms, and that pattern seems to be fairly general. So it may be that female mutation load is reduced by this sexual selection against males, and that could help sexual populations compete with asexual populations, for example, sort of partially offsetting the cost of sex. This also reminds us that sometimes sexual selection can aid rather than oppose natural selection and could act on any locus in the genome. Okay, so I'm going to turn now from the effects that mutations have on fitness to sort of the opposite. How does mutation rate depend on genetic quality? So I want to start this part by just thinking about why a mutation might arise in a given individual. So the probability of mutation will depend on that individual's ability to replicate its DNA and effectively perform DNA repair and prevent DNA damage. And of course, we know that there are specific genes that are directly involved in those processes. But we can also imagine that the condition of an individual could play a role in how effectively it will be able to maintain its genome. And by condition, I just mean the pool of energetic resources that an individual has available to allocate to various traits. So for example, if we rear flies in crowded conditions, they grow up to be really small, and we would refer to those flies as being in low condition. Another reason, though, for low condition might be low genetic quality, meaning that there are deleterious alleles present in the genome that reduce that individual's ability to acquire resources. Well, if that's true, that means that genomes of low quality could be maintained less effectively. One way of looking at that is that the mutation rate is actually not going to be the same for all individuals in a population, and it could also lead to a positive feedback loop of mutation. There was some suggestive evidence that this type of thing could be going on, but I wanted to test it directly. So again, I did a mutation accumulation experiment with some sort of novel modifications. So now we're going to look at both chromosomes 2 and 3. Again, these are both autosomes. Second chromosome looks like what I described before, so the blue copy is where we're going to focus on the accumulation of new mutations. We also have some recessive markers that help us track the chromosomes. And then this chromosome I've marked in red is special, and I'll call it the treatment chromosome. In some cases, it was wild type, and in some cases, it carried one or two deleterious alleles. Regardless of what's happening on that treatment chromosome, we maintain the lines in the same way. So a male with this genotype is crossed to females with recessive markers, and a single son is chosen of the same genotype to start the following generation. And again, males lack crossing over, and so those two chromosomes should be transmitted intact through the generations, and they shouldn't change unless there's a mutation. So in one group of lines, that treatment third chromosome was just wild type. I'll call that the high genetic quality group. And then I had 10 other groups of lines where that third chromosome carried one or two treatment mutations, I'll call them. So what those are just marker mutations that are commonly used in fly genetics. We know that they reduce body mass, which is our measure of condition here. We know that they reduce fitness, but as far as we know, they don't play any direct role in DNA replication or repair. So these are meant to represent generic deleterious mutations. Okay. So then once mutations have accumulated, I want to see how fitness has changed in the lines. So first I'm going to do some fly crossing again prior to that fitness assay. One thing we want to do is make those focal chromosomes homozygous to test the homozygous effects of mutations. But another thing is we need to get rid of that treatment third chromosome because it's carrying deleterious alleles that we already know affect fitness. We're not interested in their effects here. We're interested in their effects on the mutation rate. And so we do crosses to remove them. Okay, this is about 380 mutation accumulation lines, and again, about 50 generations of mutation accumulation. Okay, so now let's look at how fitness changed in just the high genetic quality group first. And again, this is just a measure of fitness, the fitness of those focal chromosomes in the absence of the treatment chromosome. Treatment chromosomes have been removed when we collect these data. Okay, so fitness is going down. That's what we generally expect. Mutations are accumulating that are deleterious. But the rate of fitness decline was a lot faster in those 10 low-quality groups of lines. And that suggests that those treatment alleles were causing some increase in the mutation rate on a totally different chromosome. So this was pretty exciting, and it was the first direct evidence that that dynamic could be taking place. And I'll talk about this data a little bit more, but first, I just want to think about what the possible mechanism behind that effect might be. So one possibility is that genomes that have deleterious alleles in them could be more susceptible to the activity of transposable elements. If they're hopping around more in those genomes, that could lead to more mutations. It could be that low-quality genotypes use lower fidelity DNA polymerases, and so they're making more errors during DNA replication. Or there could be some other difference in the way that DNA is damaged or repaired between those groups. So to try to get at some of these possibilities, I got genome sequence data from a subset of the mutation accumulation lines. This is paired-end Illumina sequencing. I had 112 mutation accumulation lines pooled into 38 samples. And so what I want to do here is statistically compare the mutation rate between groups. It's not sufficient to say there's 10 mutations here and 20 mutations here, because we could very easily have a difference in our power to detect mutations between groups, for instance, due to variation in coverage. In addition, it's possible to get false positives pretty easily due to mapping error and problems like that. So what I did was set up a custom pipeline where we're strictly eliminating false positives and using strict criteria to call new mutations. And then I'll use my criteria to assess what my power to detect mutations would be at each site in the genome. And so all the data I'll tell you about have been corrected for our power to detect mutations. So even with those pretty strict criteria, we got a really large data set of new mutations to look at. So I did find some differences between the groups, but only for some types of mutation. So for single nucleotide substitutions, the rate was the same. There's no evidence that low-quality genotypes were making more errors during DNA replication. Transposable element insertions, we detected a number of these, but the rate was the same. There's no evidence that the low-quality genotypes were more susceptible to transposable element activity in this case. What I did see was that the rate of small indels was about 2 times higher in the low-quality genetic backgrounds. And so that could be responsible for the difference in the rate of fitness decline that we observed. We definitely think that indels are much more likely to be deleterious than single nucleotide changes are. At least in these data, they are much more likely to change protein sequences, for example. There's also no evidence that selection against mutations occurred in the experiment. I infer that based on a couple different tests. So for example, we can see whether mutations were less likely to appear in genes than we would expect. And there's no evidence that selection was acting. So the mutation accumulation process was working as expected. I also saw something that's not normally possible to detect in mutation accumulation experiments. When we looked at these focal chromosomes that I've been showing in blue, we found these areas where there were a number of derived changes, so alleles that were different from the ancestor, clustered together, that matched polymorphisms that were found in the population of flies that I used to maintain the mutation accumulation lines. So these gray VG chromosomes there. I would find that in one mutation accumulation line, but not in any of the others. So what's going on here? These are gene conversion events that were occurring in males, probably in mitotic cells in males. And these happen when there's a double-strand break. So if there's a double-strand break in a cell, it has to be repaired in some way or the cell's in trouble. One way to repair a break is to chew back the broken ends a little bit. Those broken ends seek out matching sequence, which they can find on the homologous chromosome, and they copy from it to repair the area around the break. And so this is what we're seeing in the mutation accumulation lines, and it's pretty fun that we can see those happening spontaneously. But what's even cooler is that the rate of gene conversion events was about 3 times higher in the high genetic quality background. So we have this contrast here, where on the one hand, low genetic quality seems to increase the rate of indels, and it also seems to decrease the rate of gene conversion. This is interesting because indels can also be the result of a particular kind of double-strand break repair. One way to repair a break is called non-homologous end joining, and it basically involves sticking the broken pieces back together. When that happens, though, you get an indel, an insertion or deletion of new sequence. And as you can imagine, that's a relatively fast but not a very conservative way to repair a DNA break. Again, when those indels occur in genes, they're probably going to cause problems. So this is in contrast to the homologous repair pathway that results in gene conversion. Homologous repair is slower because you have to go out and find matching sequence to copy from. But it's going to be more conservative because on the one hand, you're not changing sequence length by doing that. Plus, the matching sequence that you're copying from has already been screened by selection. So mutations aren't necessarily being introduced through this process. So we think that the contrast here between indels and gene conversion means that flies of low genetic quality were using this less conservative DNA repair pathway, and then the good quality flies tended to use the more conservative pathway. As I said before, we don't think that the treatment alleles that were involved in the experiment, we don't think that they play any direct role in those DNA repair pathways. Based on their annotations, they shouldn't. It's unlikely that all 10 of them would. Rather, we think that what the treatment alleles do is reduce overall condition, and that in turn leads to a difference in the way that DNA is repaired. So now I'm going to show you more evidence that makes us think that that's the case. I showed you before that fitness declined more rapidly in those backgrounds of low genetic quality. But you can see that effect is obviously pretty variable. Those slopes differ quite a bit. It turns out that a really good predictor of the rate of mutational fitness decline during the experiment was the body mass of a fly with those treatment alleles prior to the experiment. Okay, so in blue up there, that's the high genetic quality or wild type kind of fly. It's the heaviest one. It has the best condition. And then condition goes down to varying degrees when we put treatment mutations in. When body mass was reduced to a greater extent, we saw a much more rapid rate of fitness decline. So that definitely suggests that condition is what's mediating this effect. These data are a little noisier, but we did also see that body mass was a significant predictor of both indel and gene conversion rates. And so again, that high quality fly, or the high body mass fly on the right, has more gene conversion events relative to indels, and then the reverse is true for the low condition flies. Okay, so what we think was happening was that genetic quality influenced condition. So the presence of deleterious alleles reduced condition, and that led to a difference in the way that DNA was repaired. A lower fidelity repair pathway was used to fix double-strand breaks. And that resulted in more indels in those low-quality, low-condition genotypes. And so that could lead to a positive feedback loop where genetic quality sort of depends on itself. That could have a number of implications for populations. So one thing that suggests is that in a novel environment where condition may be reduced, we should see greater mutational variance because more mutations will be happening. Choosing a high-quality mate might be a good idea, not just because that will mean good genes for your offspring, but also because it might mean fewer new mutations in your offspring. In sexual populations, the extra mutations that are generated through condition-dependent mutation get mixed into the rest of the population through sex. And so it turns out that condition-dependent mutation will reduce the mean fitness in sexual populations. However, it's also true that it's thought to be the case, based on theoretical modeling, that condition-dependent mutation should accelerate the rate of mutational meltdown in finite asexual populations. I also want to note that although I've been talking here about mutations that are occurring in the germline, the same DNA repair pathways are going on in somatic cells as well. And so it's possible that these results are relevant to the rate of cancer-causing mutations, for example. OK, so now I'm just going to tell you about a couple more types of variation in mutation rate that we observed. It's now been seen several times in a few different organisms that the rate of substitution varies with local sequence context. So here in this plot, this just shows each of the possible 3 base pair contexts, and the mutation rate for substitutions in that center base varies quite significantly. This was not associated with condition, but with location in the genome. We also saw that indels were more likely in regions of the genome with low GC content, and that gene conversion events happen more often further from the centromere. Now, we don't understand the mechanistic basis behind those patterns yet. But as I said, we're observing things like that in multiple taxa. One thing I find interesting about that is that some of those patterns do seem to be consistent among different organisms, and other patterns are not consistent. And I think we really need to figure out why. One reason to be interested in this kind of data is that it should inform more detailed models of molecular evolution. There's also growing evidence for differences in mutation rate among what you might consider to be strains, or different genetic backgrounds of a given organism. So here are 5 sequencing-based estimates of the substitution rate in fruit flies. And you can see there's some evidence of variation there. I might have said that this variation could be caused by different analysis methods. So people are maybe analyzing the data in slightly different ways and getting different mutation rate estimates. But I don't think that's the whole story, because these 2 blue values here, they come from a study by Schreider and colleagues. And they looked at 2 genotypes using the same methods. And they find a 2-fold difference in substitution rate between those groups. Those guys on the high end have an elevated rate of GC to AT transitions. And it turns out that the estimate we got is intermediate. And indeed, the spectrum of mutations is also intermediate, with a somewhat elevated rate of GC to AT transitions. They speculate that there could be a mutator allele here, but no one has found what that is yet. OK. So I hope I've convinced you that mutation can actually be quite a variable process, and that some of that variation is systematic and predictable. So if we think about how mutation happens in a given individual, I think it's fair to say that it does depend on traits in that individual, for instance, DNA repair traits. And what that means is that we can treat mutation as a trait. Just like any other trait, we would expect it to be susceptible to variation in genetic background, differences in the environment, as well as to evolve by selection and drift. So I think we need to examine what we mean when we say this is the mutation rate for organism X. Do we mean it's the average mutation rate across genomes and conditions? And if so, how many samples do we need to be able to get an accurate estimate of that average? Perhaps a more interesting question is, what is the genetic variance in the mutation rate, and what is the mutational variance? This kind of variation, I think, matters, partly because the evolution of genomes is intrinsically interesting, but also because it has theoretical implications for how we expect populations to evolve, and practical implications for how we analyze sequence data. People are looking at the published genome sequences that are out there to address a number of interesting questions, and obviously I think that's great, but I do want to point out that there are some kinds of mutation rate and effect variation that we won't be able to detect unless we do experiments. And so in that vein, I think that the combination of sequencing technology and the mutation accumulation approach could be a really valuable tool in that endeavor. Okay, thanks very much. Do you want me to take questions? Or?
Maybe somebody has one question?
I could ask a question, actually, Nathaniel, in the middle. Hey, great talk, thanks. I'm wondering if you had any thoughts about a mechanistic link between condition causing mutation, causing the double strand breaks and then causing the indels, because I was thinking potentially lower condition individuals might produce greater reactive oxygen species or hydroxy radicals, and we know that those cause double strand breaks in the experiments that we do.
Yeah, so we looked for a difference in the rate of breaks between the groups by just comparing the total number of indels plus gene conversion events in high-quality flies and low-quality flies, and there wasn't a difference there. So we would infer that the breaks are happening at the same rate. It's just that the way they're repaired is differing. So I might speculate that something to do with cell cycle, the timing of cell cycle, because one pathway takes a lot longer, right? We should look in that area for a connection. Thanks again.
Thanks.